So I assume everybody can see the screen now, and this is the presentation, and we have a few slides. Now today's topic is COVID-19 and BME. It couldn't be more timely. The whole world knows about this coronavirus disease, 2019. Now, the reason I raise this topic because of its relevance to BME, indeed, it's very relevant. Now, on this slide, you can see that at the bottom, we've got the artist's impression of the actual virus. So it's called coronavirus because of the shape it appears. It's got all these things sticking out, round in shape. But at the top here, top right corner, you can actually see the real thing. This is actually the electron microscope image of the real thing of a coronavirus. As you can see that it's actually, all these things sticking out. Now, just browse through the internet, you more or less can see this information, right? There is a daily update of figures reporting how many people actually get diagnosed positive and how many people actually die because of that since January this year. So in the past 5 months, we have just over 5 million. Now, in fact, the data was captured on the 22nd of May, about a week ago. Now it's gone actually higher than that. And 5 million plus worldwide diagnosed. Then about half of them recovered or case closed. And then among those case closed situations, we have 334,000 passed away due to this disease. And it's very interesting because you can see a very low rising rate for the first 3 months, and then suddenly everything starts to blow up, and then you've got very sharp increase, very steep slope. Now, coming back to BME or even university education, I try to inspire discussion. Now, I've actually listed out some of the key elements of what university education is about, particularly BME. Now, students, when they actually joined our program, we always remind them these are the things that they should acquire. Okay? They should actually look at facts in front of them. So for us today, we look at the facts in front of us concerning COVID-19. Okay. Now then we start to understand those facts. We try to correlate our observations with some of the underlying parameters like culture, like international politics, science and mathematics and so on. So you can more or less immediately see those facts and then use these aspects to analyze those facts. And then from those facts, we learn from them. We learned information, we learned knowledge. So once we got those knowledge, then we can apply them. And this is exactly what BME is all about. This is what our students, throughout 4 years, we train them to do, day in and day out. Now, for today's forum, it's very short, only 30 minutes, but we are prepared to actually extend it to about an hour if there are many questions we need to answer. So now you can type in your discussion question, and then we reply in the message box. Okay. Now, Professor Megan Ho and Professor Duan are the 2 members of the forum here who actually do your try to tackle your live questions we receive through the message box. Okay, this is the first time we ever done this exercise. We see how it goes. Okay. Now come back to the data. Now I've shown you the 2 plots One is the number of people, number of positive cases. The other one is actually the number of deaths. Now, if you actually look at how diseases get spread and transmitted, there's a very well-known model. It's like a population. You get number of people get affected and then transmitted to how many people. So that's how many per patient get affected. And then the number of cycles, so how many generations of transmission, N, capital N. So A to the power N. If you actually plot that, then you can actually see that it's like an exponential growth. In this case, it's actually the red line. So the plot we see for the number of positive cases ought to be modeled by this simple y equal to A to the power capital N. However, the reality is not. It's slightly different. You've got a flat starting stage and then the steeper later stage, quite linear. Now, why? Well, we can think of possible reasons. For example, lockdown measures, because governments and everybody start to alarm people, and then people actually don't go out. So that stops the number of people get affected. So it slows down the A to the power N process, and then people actually get more aware of the transmission pathway, and then they start wearing face masks, and then that again modified that plot, and that's why we ended up this rather skewed plot. So in other words, you can actually see that if you actually use mathematics and you actually put in those parameters, you might be able to, or sooner or later you can, model the progress of diseases by looking into other factors, taking into consideration other factors, wearing face masks, and lockdown measures and so on. Now let's look at the death rate. Now, quite good correlation, and you see that the blue line correlates the brown lines very well. So in other words, 14% death rate is quite robust, reliable. It's a linear proportion. Okay, but towards the later stage, more recent weeks, it starts to slow down. The correlation starts to become less because people have more experience. Doctors know how to treat them. Doctors know how to prevent the complication, and also invention of new treatment strategies and so on. So as you can expect, if actually time goes on, that plot, that brown line will actually start to tail off, and then the 40% will start to drop. But this is what we hope to see, actually. So in other words, mathematics itself is already a very good knowledge that we can learn. BME is something that basically helps students to understand those facts. Now, BME also helps people to generate new devices, new instruments. For example, this thing which only released in April, specific for the detection of COVID-19 viruses, and it actually can be sample to answer in 5 minutes. Because of the very big, enormous market need for such devices, then this company called Abbott, which is very famous, company for manufacturing such instruments, put together engineering resources, and then immediately they actually released this rapid diagnosis instrument kit. Okay. Now, if you actually look at the entire healthcare system for any kind of diseases, including COVID-19, you have prevention, and then you have diagnosis, and then you have treatment. Now, these are the images that we actually can see reflecting the 3 aspects of healthcare. So prevention for COVID-19 is basically aerosols. So somebody coughs, then all these aerosols start to propagate, and then people get affected. And then if you want to remove that danger, you wear a face mask. Then if you want to actually monitor, screen out people, you use the fever, temperature, body temperature. So you've got thermal imaging. So everything relates to some kind of instruments and devices that can help us to achieve prevention. In the same way, diagnosis, right? So you've got doctor here, collecting a deep frozen sample. So even actually the gloves, the rubber gloves, and then the cotton stick, and then the face mask, and also the glasses are all related one with the other, BME, because we are the engineers who actually optimize those devices, instruments. And then next to it is actually a protein detector. These are the carbon tubes for doing DNA amplification. And in fact, at the bottom here, there is an instrument for detecting DNA, which is actually what we call DNA amplification kits. For the treatment, you've got the operation theater, you've got the hospital beds, and you've got the gear for the medical personnel to protect themselves and so on. So everything, one way or the other, related to devices and instruments, and that's what BME is supplying all along. Okay, so we are the enabling discipline that supports all the instruments and non-human requirements. And value chain includes research, design, manufacture, sales, and services. These are actually the career pathways that our graduates enter. And then the expertise that we teach, international standards, science and engineering, IT and data analysis, materials, devices, and equipment. So it's very broad and it's very specific because it addresses those three aspects of the healthcare system. So how about our faculty members? How about the people in our department dealing with such transmission, infectious diseases? Jonathan Choi, one of our professors, is actually researching into nano materials. Nano-sized materials can easily go through the cell membrane and go directly into the inner organelles of cells, and then we can start to engineer those nanomaterials to make them effective, make them functionalize in treating diseases or inducing biological effects. Now, Jonathan Choi is a very special case because he's actually one of the top high school graduates in Hong Kong. He actually received 10 distinctions, and then he didn't go to medical school. He went to BME. And then the next one is Professor Megan Ho, who is actually right next to me, and she develops a fast diagnosis kit. In this case, it's actually a platform which is made from cellulose paper, and then surface tension can actually spread the sample, and then they get functionalized with nanoparticles or proteins, and then protein will trigger a reaction, and then the target proteins will actually, the target ones, their presence will trigger a change of color, because gold nanoparticles have different color. So very simple point of care device. And then Professor Megan Ho actually has a quite broad profile of research topics covering, in this case, the first one is DNA Nano Sensor, which is a technique that generates circular dumbbell DNA sequence. And then these dumbbells DNA sequence generated through some kind of special enzyme, and then they can actually help to amplify the target DNAs. And then she's also specialized in using droplets as a reactor chamber. As you can see in this video animation, actually video, two droplets containing two different contents can be sent to merge together so that a reaction can take place. And using droplets has the ability of very small sample quantity, and you can program those droplets to do whatever you want. And then Megan also looks into separation of organelles and cells using a spiral microfluidic channel that can actually, through this action, when solution goes through the channel, will be split into different contents according to their weight. And then she's also involved in artificial tissue, using pocket cells to construct man-made tissue structures. And then finally, it's my own work. I've been developing a device called lab on a disk, using centrifugal force to actually spin and spread body fluid, like in this case, blood, droplet blood, and then it has actually get spread along a channel radially. It actually goes through reactions to get amplification of DNA and to get detection and so on. And the beauty of this is actually it's very disposable, very low cost, and you can actually do multiple tests on the same device. And my job is actually to integrate all these processes in a single printed circuit electronic board, as you can see in this video clip that such a device is in operation. This particular one is actually for detecting genetic modifications in fruits, particularly papaya. So that's so much about my presentation, COVID-19 and BME. Now, I would like to invite questions to do our best to answer. Okay, so now the time is actually head back to delegates. So do we have any questions? Can you unmute?
Hello, sir.
Hello.
Sir, good afternoon. I am from India.
Hello. Wow, you're from
Sir,
India.
thank you, sir. Sir, I am having one doubt regarding coronavirus.
Right.
So, recent news I am seeing here that coronavirus is changing its structure, color, everything there. From the news, I am knowing this. So, I need to know regarding this issue, sir. How it is changing,
Right.
self-mutating.
Right. Well, did you, Mr. Don has an answer.
As you mentioned, the mutation of those virus in different regions, is that
Yes.
is that question? Yeah. So you're asking about the mechanisms of the mutations, right?
Yes, ma'am. Yes, ma'am.
Okay. So the RNA mutations change every day. So anything can be the cause of mutations, like the UV or the environmental stimulus, like some chemicals. Any perturbation may cause the RNA mutations. But the difference is that some mutations can be so effective, can be so important that it can be heritage, but some mutations may be lost. And then in that sense, we can call another process called evolution. So there may be some selection process that specific mutations can survive.
Okay, ma'am.
And can lead to a significant number that people can detect. So all of this, so you can kind of see all this sequencing or all these mutations people can detect actually rely on one very important technology Professor Ho just mentioned. It's called the DNA or RNA sequencing, the nucleotide sequencing. And then I would like to correlate back to BME, how BME play a role in the DNA RNA sequencing. So the technology, so the DNA sequencing technology can stand back maybe from 30 years ago, 40 years ago. But during the last 40 years, the speed of sequencing increased more than 10 million times or even more right now. So that's why when, for example, before if we want to sequence a human genome, it may take like tens of years. But right now, if we want to sequence genome, it takes just like tens of minutes or just several hours. And then if you want to sequence specifically the virus, the new RNA, just take less than 1 hour. And then BME play a very important part. We can design as an engineer, BME engineer, can design the sequencing machines. We can optimize all the process, and then we can engineer the enzymes in it. And then coming back to BME in CHK, one thing I would like to bring up is in the first year, we offer a course, laboratory course, that enable every student to design a machine that can carry out those sequencing reactions.
Okay, ma'am.
So,
thank you.
Yeah, so in the end of this class, in the end of this course, you will have your own PCR machine, the sequencing machine, and then you can design your own manual to tell other people how to operate this machine.
Okay. Since I'm not belong to biomedical engineering department, I am from electronics and instrumentation engineering department. So the scope of the course is scope of BME in COVID is the title. Right. I need to know what is the scope of instrumentation in biomedical,
So let me address that then, because I actually come from the electronic engineering background. I actually spent 20 years in the electronic engineering department before I joined BME. Now, the instrumentation side of it, let's take COVID-19 as an example. Okay. Now, if you actually look at the history, when the first few cases was identified, what did they do? They quickly sequenced the DNA, the RNA they recovered, okay, like Professor Duan just mentioned. Now, in fact, sequencing DNA nowadays, it's sort of very fast, requires a lot of sample processing. It requires a lot of detection of light, particularly fluorescent light. Okay, so in my case, I developed devices to detect light, to actually generate temperature cycles, to split those DNA, then ask them to amplify. Okay. Now, again, you mentioned about mutation. Now, these viruses mutate very quickly, so they change all the time. Now, how can we cope with those changes and be able to develop them, or develop a vaccine or a technique to detect them? We also need to change. So from time to time, we sequence those virus DNAs, require instruments, and from time to time, we develop new instruments to detect them so that the cycle time is very short. Now, if you take, for example, influenza as an example, every year there is a new type of influenza, and then the vaccine will be different. So we constantly sequence, we constantly develop new vaccines, all because of the instruments that we made that provide us the fast turnaround data. Have I answered your question?
Yes, sir.
Yeah.
And another one more question, last question, sir.
What?
One more last question.
Okay, please.
Sir, that is in instrumentation, we are studying about sensors and transducers.
That's right.
In biomedical analysis part, which that is, I need to know the applications of sensors and transducers.
Right. Well, the applications,
analytical instruments.
Now, let's take, for example, protein as an example, okay? Now, we detect protein all the time because, the body generates a lot of protein, and each protein has a specific binding to another protein. And the presence of those proteins is important. Now, how do we detect them? What kind of sensors are we talking about? Well, in fact, there are many. One, we detect weight. Each protein molecule has a weight. Let's multiply it by 10. So 10 protein molecules is usually equivalent to femtopicograms. Okay, femtopicograms. So we detect sensors that detect pico or a femtogram change of weight. Okay. What do we use? Quartz resonant resonator. we use quartz, detect acoustic wave. Other times we detect light. The presence of certain protein can change light intensity. So by measuring change in intensity, we know the presence of certain protein. Now there's a very famous type of device called bacteria sensor. It's very widely used. Okay, people actually go to a doorknob or go to some handle to see if it's clean. They have this thing. What did they do? They actually take a sample from the surface, mix it with another chemical enzyme, and then when they react, they generate light. Because living cell has ATP, and ATP react with those enzymes, generate green fluorescence. So we turn live bacteria into generation of light.
Okay.
Photons. Okay? And so,
Okay,
right?
Okay, so thank you for your information.
Right, you're welcome. My pleasure. Look forward to meeting you here in Hong Kong. Right. Now, I have one important question that I received from one of you. It's actually asked the difference between bioengineering, chemical engineering, and biomedical engineering. Well, if I have a live class in front of me in the lecture theatre, what I normally do is I'll throw the question back to the audience, and I'll pick a few of you and answer. Now I can't. What do I do? Should I pick one of my colleagues to do it?
Maybe we can try to see if anyone would like to address that question.
Yeah, anyone would like to address that question?
In your opinion.
In your opinion, right? Yeah. You can type it in if you want.
Or you can raise your hand. Would you have a raise your hand function here?
Not really.
Okay.
Not really.
Well, in that case, I'll improvise. I'll actually answer the question. Bioengineering, most of the programs that I see, although they overlap with BME a lot, more concerned with mechanical aspect of the medical side. Okay, they're concerned with mechanical aspect, like they deal with the physics, they deal with the material strength.
At the molecular level.
Yeah, at the molecular level, yeah, not the real mechanical engineering. And then chemical engineering is more broad. Chemical engineering deals with, for example, color, paint, how certain chemical reactions get used. In fact, for example, oil refinery. For example, from crude oil, what kind of products can you generate? And how to treat services and make them, certain become more functional. Now, biomedical engineering is more specific with dealing with healthcare, hospitals, equipment that are used in hospitals, and the needs of human healthcare, treating diseases, detecting diseases, operating theater instruments. A lot of it is electronics, MRI, X-ray, ultrasound, even implants, artificial implants, and so on. So more of the instrumentation, and although we also deal with molecular stuff, which actually overlaps with bioengineering.
I agree. Yeah. But also, I'll just add one little point that biomedical engineering perhaps bringing a little bit of the perspective from the hardware side. So sort of develop the hardware, like what Professor Duan mentioned. You will have a chance to get hands-on experience to develop a PCR machine based on the sequences provided by our colleagues in the medical field or in hospitals and so on, and sort of impose this information into the machine that we're developing right here. So it's more or less solving the medical issues utilizing hardware.
I hope I've answered the question. If I have, then let me know. If I haven't, also let me know.
So we have one question. I think this is from Costi, the gentleman that we talked with in India, asking about the content information about research guidance. I suppose we have that content information on the slide.
What should I do? Share the screen?
It's on the last slide. We have the content information.
Oops. Can't give up. This one? This one?
Yeah, suppose we have all the content information on the last slide, including the website.
What happened?
Previous. Yeah, here we go.
There you go.
There's the website and our Instagram, Facebook information where you can actually find all our content information. Maybe give you some time to take a picture or screenshot.
Yeah, take a screenshot of this. Great. Any other questions?
I have one question. So I was asked if CRISPR is effective in treating coronavirus. This is really a good question.
Yeah, this is a good
are
question. Yeah.
at the forefront of BME. So CRISPR can be a potentially very promising treatment way for many diseases. So there are a lot of startup companies are working on it, and they're pushing forward CRISPR as a research tool to a real therapy for patients.
Yeah, once the green light of certain therapy is it's there, then it will actually explosion of such treatment.
That's right. That's right. Because once we want to change the DNA level, it involves a lot of ethical issues. So in terms of gene therapy, so we do have several gene therapy on the market, but right now only just several. And then the first gene therapy in the world on the market emerged only 3 years ago. So CRISPR is on the way, and then many of us believe it has a bright future.
I can imagine that actually CRISPR will find its way first in treating cancer, because cancer is more sort of imminent, and for a lot of terminal cases, there's not much to lose. CRISPR will be much more effective.
Yeah.
Any questions?
Any other question?
I see that we have a very quiet crowd.
For this one, let's see. Sorry. Participation easily this program. Oh, for this particular event, I wouldn't think there is any e-certificate, because it's basically just like a lecture or an information session. So e-certificate, probably we don't regard it as necessary.
Another question is about the curve.
Yeah, the curve.
Equals to a to the power of n. The question is about the presence of lockdown measures and face masks. Although the curve seems to reach the observation criteria is that those curves reach their peaks with almost the same value on almost the same day. So the question is about why it is the case. This preventive measure could potentially reduce the number of transmission cycles.
Well, I actually draw those curve, although I actually use the y equal to a to the power n function to draw that curve. But the two actually meeting up towards the end is artificial. It's not a very accurate description, I must say. So I wouldn't actually recommend anybody to actually take that convergence seriously. And I think the message that I want to get across this particular case is that there is a definite kink for the blue curve, and that kink is not there for the red one, right? So if it's a natural, unperturbed situation, there won't be any kink. But because it's a perturbed situation with all this lockdown and face mask situation, it distorts. The curve has been distorted, and that's the message. Now, in fact, I'm actually oversimplifying the situation by simply, suggesting possible reasons. But what I'm trying to say is, in fact, if you know your mathematics well, if you know how to analyze data, which is actually what I try to convince everybody to be careful, is those 4 principles that every one of us should actually learn. It's a lifetime, it's lifelong learning. That we look at the facts in front of us, we understand those facts using scientific analysis, and then we correlate those facts to things happening around us and to scientific principles, including mathematics. Learn from them and apply. That's what we all do. Myself, at least, anyway.
Yeah. We have another question. It's very specific. I suppose I'll put it down as a very general way. So the question is about what if I do EE or other engineering major as a bachelor degree and then do BME as a master's degree, versus perhaps choosing and BME as the major to do the bachelor training as opposed to others.
I think we probably have different answers.
Yes. Because we all come from different backgrounds.
Yeah. I mean, I must declare, I come from a very strong double E background. And then I switched over to BME.
I'm from mechanical engineering and then switched to BME.
And I'm from chemistry, not even engineering. I'm from science part, and then come to BME right now.
So it depends. It all depends on what a student wants. Right?
Yeah.
Some may actually want to do BME first and then double E for masters. Now, I think the two programs, the EE and BME, have different emphasis, very different flavor. And although they deal with, some of them, the topics are overlapped a lot. BME, I can say, if you read our syllabus, our course list, you know that it's a combination of many things. It's very broad, honestly. It talks about, fundamental science, and then a lot of healthcare-related topics, and then a little bit of instrumentation, including electronics. But I would say if somebody who is interested in pursuing a career in the healthcare industry, I wouldn't see any reason why he should not do it first. Because that is the ticket for you to get into the profession. If I was an employer in the biomedical industry, I wouldn't consider a BME graduate more than a double E graduate. So that's basically it.
Well, my opinion is that it's the problems or the areas that you're interested in the most matters. So, for example, if you're interested in biomedical issues or biomedical problems, I agree with Aaron that perhaps BME first will sort of satisfy your desire to understand what are the questions out there. But eventually, down the road, solving the questions, you may need to collect information or collect knowledge from electronic engineering, mechanical engineering, chemistry, physics, and biology in order to address that question. So it really matters in terms of how you would like to approach that problem. Do you prefer to collect all these fundamental information before you immerse yourself in the biomedical setting, or would you prefer to immerse yourself in biomedical settings before you sort of narrow down the knowledge relevant to the question? And it's from my perspective. But from engineering side, perhaps.
Yeah,
It's completely different.
yeah, you probably have a different.
So my personal experience is a little bit detouring because I'm from chemistry part, a science part. So I initially as an undergrad, I don't know where was my interest. So I just explored, and then during my undergrads, my major is in chemistry, but I did a lot of materials, science, and engineering stuff. And then I realized that I'm more interested in bio. So I did my PhD in chemistry department, but choose a more bio stuff. But after several years, 6 years PhD training, I found myself more passionate about real healthcare and medical questions. And then I switched to biomedical engineering. So if you really know that you are interested in medical questions, you more care about the healthcare issues, health and life questions, I will encourage you from the start, focus on medical related, for example, BME.
May I invite Yuki, you to share with the audience the expertise, the research topic that you're now, pursuing. I think we probably, I think the audience is interested.
Okay. Yeah. So I got my PhD in Stanford University, and I joined Stanford in 2010. So some of you may know the word called optogenetics, so which is developed by a professor in Stanford in 2009. So the concept is that use light to control cells and animals. So I got very fascinated about this idea. So and then I did my PhD work in Stanford and postdoc in Stanford and bring things back in BME. So what right now our group is doing is to use genetic and optical approach so that in the future we may develop a therapy that can use light to treat diseases. For example, if you have something like skin diseases, just shine some light, blue light, red light, and then gradually it can be healed. Or you have some disease even in the brain. So that's actually what people is doing right now in other groups. For example, some people will feel like depression just by deliver some light into the brain, and they can treat the depression. So this is what we are doing. So we're developing using those different approaches, and then BME concept to use light to treat diseases right now.
Let me share a little bit more. I remember seeing one of Li Ting's video she produced. They implanted some light-sensitive brain cells into the mouse. And then the mouse actually, when you shine light to this mouse, the mouse goes to one way. And then when you actually switch off light, the mouse goes to the other way. It looks like you can actually control the motion of a mouse by switching light. Am I correct?
Yes. So the basic method is we first genetically modify the neurons in the animals so that the neurons can be controlled by light. And then theoretically, any part of the neurons, any kind of neurons can be modified, and then any neurons can be controlled. So not only we can use light to control the behaviors, the movement of animals, and the mood of the animals, whether they are happy or they are depressed, can also be controlled by light. So it is indeed a very fascinating new technology right now. And
So maybe I'll ask follow-up questions according to one of the student Jeannie's question. So Jeannie is asking whether or not whether the courses of BME emphasize more on biological knowledge or on engineering techniques. So I'll use that question to ask to invite Professor Duan to introduce the biological knowledge and engineering techniques utilized in your research. to give you some flavor.
Some flavor. Yeah. So biological question knowledge right now, in terms of what we are doing optogenetics, we definitely need to know the basic molecular and then cell level of the neurons of the cells, and then we need to know genetics. And then in terms of engineering, we need to do genetic engineering to change, to modify the behaviors of the neurons. And then because it's optogenetics, we involve the delivery of light. So we may combine with mechanical engineering and electrical engineering to see what's the best way to deliver the light in it. So it's quite interdisciplinary. A lot of knowledge can be combined together to focus on one specific BME question.
Yeah, so short answer to Jeannie's question is that you're actually going to receive educations on both about biology side or medical side and also engineering side. As elaborated by Professor Duan, that the engineering concept in BME is not only the hardware instrumentation, not only electronic engineering or mechanical engineering or computer engineering, also at the molecular level, genetic engineering, protein engineering.
Yeah,
so
bioengineering.
Right, so it's really across different landscapes.
So in the first 2 years, we have the chance to get different aspects of bioengineering, and in the last 2 years, you can choose which stream, which more detailed, more specific direction you wanna study. And in so in the last year, we provide a final year project, and you can choose any group, any professor in BME or even outside BME department to do this project. So if you, for example, if you want to do more biological engineering thing, so we have different choices. And then if you want to do more mechanical engineering or electronic engineering, we also have different choices. Yeah. So we do provide a lot of possibilities for you to choose and develop your own strength.
If I was to add anything from what I've observed, BME students, if they want to do well in their first year foundation courses, which addresses basic science, including biology, chemistry, physics, and mathematics, and for the biology side, they have anatomy and also life sciences. If our students can do those courses well, it's going to be much, much better for their senior years. Because, I mean, in the senior years, they have image analysis computing, nanotechnologies, chemistry, right? Then the instrumentation is physics and a bit of engineering. Then biomechanics is a bit of mechanical engineering and material science. And they're all founded on science and maths, right? Yeah, that's how I say. Now, we have another question, which is very interesting. Now COVID-19 has generated a situation that there are many disposable masks. People actually get rid of them after they finish using them. I mean, I already got one and I'm going to throw it away afterwards, right? Now, it's going to create a lot of problem, right? Because we have all these disposable face masks. Well, I don't have an answer to that, how to deal with them. But in my opinion, a small face mask of that kind, the amount of quantity of material that people actually put in using them, is not a lot. Now, surely you cannot just throw them into the sea or into the river, but you can actually, they are quite inert, so they can be sort of in the landfill material. They're not biological waste, right? But on the other hand, they are creating some waste material, but the mass is not very heavy. So compared to, for example, the plastic bottle, the amount of plastic that this one carries is probably more than this one. So pollution-wise, I guess it's not a big problem. Rather, the manufacturing process involves energy. It takes more energy to manufacture this. But then it's a fact of life. So disposing them, it's up to anybody's choice. But making reusable ones, which is what our government is trying to do, is probably one possible ways of solving it. I don't have an answer.
Yeah, I don't have an answer. That's a tough question.
It's a very tough question.
Perhaps a question to your generation is addressed.
Yeah. Making very light ones.
Yeah.
Very light ones, and making biodegradable ones, right? And that's a lot of people actually want to do for plastics, biodegradable plastics. Perhaps get rid of diseases altogether, never have to wear any of those, right?
Or something that totally changed the way how we wear masks, right? So perhaps just a small size of fiber covering the nose and ears, like the earphone, right? So it's getting smaller and smaller. You just need to just
And I noticed this is very tiny air purifier, like this big, that people can have on around their neck.
Yeah.
Maybe in the future, your generation can develop something similar to the purifier that can kill all the viruses, like 20 centimeters away from you.
There's an interesting point brought up by, again, class from India. Is it possible to produce biodegradable masks in the world? So if you think about all these materials, these different layers, perhaps the woven layer is somewhat biodegradable, but the middle brown layer is perhaps the one made by polypropylene. I think that is perhaps non-visible, non-biodegradable at the moment. How do we actually generate nano-scale biodegradable fibers with the same characteristics? I think that's the challenge.
It could be possible, in my opinion, because now the approach of doing biodegradable plastics is actually add additives that will actually, after receiving certain temperature, they will start to break up, so that your polymer is no longer a polymer. It's actually sections of short chains.
Yeah.
So once that happens, then it will just degrade, and then it becomes an ordinary benign short chain hydrocarbon.
Yeah.
So you can add additives to it, but the health aspect of it, I'm not too sure. That's one thing, and the second is the engineering processability of the plastics to make it as melt-stone kind of fabric could be another challenge. Well, again, BME comes into it. I mean, sky's the limit, and there's a lot of opportunities for us to develop new innovative objects and materials and devices. In fact, what I see is the reason why I always try to promote BME is because the human problem is the fundamental need. Everybody needs help. Everybody needs good health. And therefore, the market is always there and it's always regenerating itself.
So we have another question asking about the arguments concerning genetic modification. And it's really hard to reach a consensus among researchers and scientists. So it's indeed a very important question if we want to use genetic modification in living things like animals and right now even humans. Animals right now, well, we do have different standards and criteria to do genetic modification on animals. I think that's easier to address. The real difficulty lies in genetic modification in humans. So probably you raised this question because you may have heard of the CRISPR baby some time ago. So I think the received consensus is that genetic modification should be or should not be used in embryos, or should not be used in reproducible cells like sperms and eggs, because in this case, the genetic modification will happen in every cell of your body. It's not localized, so it will affect all your part. And also, it will be heritaged to your offsprings. So in that part, we should really be careful about that. But as I mentioned before, there are several gene therapies available. So that kind of gene therapies are using genetic modification only in very localized cells. For example, the CAR T therapies, just the T cells are genetically modified. It won't affect other cells in our body, and then it won't be heritage. It won't be handed to the offsprings. Or some other eye gene therapies, only the eye cells are modified, but not other cells. So you can see the difference, whether it will be heritage, whether it will be widespread influence. So that's the key things people are trying to think before applying genetic modification on humans.
Just to add on that, I think ethical issues and regulations are very heavily discussed contents in the biomedical fields, and I think it still takes time for people to actually really set up the standard. So we actually have at least one course offered by our chairman talking about the regulation. At this point, mainly on the instrumentation side, but I'm sure the regulation eventually will be open to many different aspects of BME as well. That's what I just mentioned.
So should we stay for a few minutes if they want to ask about our program?
Yeah. Well, at this point, we still haven't received, we don't receive any more questions. Now, I'm going to actually wait for a couple more minutes. Now, if we actually don't receive any more questions, then we'll end the session. Then say goodbye to everybody, and hopefully we'll have another live session again. It looks like this one is quite interesting. We're going to be a YouTuber.
I'm not sure if I like that idea.
How can we ensure there's no side effects when we conduct a somatic gene therapy? Or else the person maybe have unknown side effects in the future? So how can we ensure this? How can we limit the side effects if we conduct gene therapy, somatic gene therapy? And then what else, or will there be any side effects, unknown side effects in the future? So if we're coming back to the gene therapy approved in the market right now, first, the eye gene therapy, why it's approved? Because in eyes is relatively isolated in environment from other parts of our body, and then it has a physical and a natural barrier from eyes and the brain. So in that case, the side effects will be limited just in eyes. So that's something really researchers are considering, the side effects. So the gene therapy is approved. So they already test the side effects, and then they are specifically just to eyes or just the CAR T cells floating in your blood. And then unknown side effects in the future. So every gene therapy before it's approved, clinical trial will be conducted for at least several years. So the gene therapy I just mentioned, the side effects, any possible side effects, have been monitored at least from 3 to 6 years. But in the long run, whether there are any unknown side effects, so this kind of question can be applied to any drug. Yeah. So that's something we don't know. But what we can ensure is that within several years, there's no obvious or very bad side effects happening.
Any more questions? So we'll wait for a little while, and then if no, then we'll close the session. Well, of course, this is not the only occasion that participants can ask questions. You can always use the email I've shown to my PowerPoint to send in your question. Our job is to actually serve students. So just drop in a few lines, and then we'll do our best to help you. Okay, right. So it looks like we've exhausted all questions. Now, thank you very much, and I hope we all enjoyed this session, and we look forward to meeting you again one way or the other in the future. And meanwhile, if you have any questions, just email us, and then we'll continue our communication. But thank you very much. So I'm going to close the session now. Okay. Bye bye.
Bye bye.
Bye bye. See you again.